Do you hear me well? Good, good. Okay, so we can start. This is really a series of webinar conducted by the Saudi Epilepsy Societies. The main theme for this to share and exchange knowledge about seizure-related topics, including pharmacy, medicine, other areas of epilepsy management. There are certain gaps in management epilepsy patient. One of them is medications, and it's a common scenarios for us that we're facing as a clinician or clinical pharmacist or general practitioners. Which brand can be used? Can I safely switch between brands? Can I safely switch between these two medicines? So for this and for other reasons, we choose this topic in the Saudi Epilepsy Society, and hopefully this can shed light on this one. We're going to have sometimes at the end of this webinar to exchange questions and comments. So the program, as was laid out in the brochure, Dr. Lina, she kindly going to start off the meeting. She's a colleague. She's a pharmacist. She's working in the King Fahd Medical City. I know her for a few years, and she's really working as a clinical pharmacist. So she's going to start off with a clinical background about the topic. So Dr. Lina, you're all welcome to start off the meeting.
Yes, welcome everyone. As by Dr. Majid, that I'm going to give just a introduction for the topic. So my name is Lina Saeed, and here I'm talking about the bioequivalence of starting with a brief introduction, then I'm going through some definitions and some facts, and then we have some experience of studies that have been comparing using of brand products of antiepileptic medication to the generic ones, and what are the pharmacokinetic properties of antiepileptic drugs that could contribute to these differences between the generic and brands. So also I'm going to talk briefly about advantages and disadvantages of using both of them, and what is the word of American Academy of Neurology about the using of generic products in antiepileptic medication. So as an introduction, as we know that epilepsy is a chronic disorder that often requires lifelong treatment, and avoidance of seizures is the primary goal, while keeping adverse effects to a minimum. And when a long-term remission has been achieved, it becomes important to avoid a single breakthrough seizure. So in response to increased pressure, healthcare systems are encouraging the use of generations, and as more antiepileptic drugs lose their patency, it is important to examine the question of what institution may problems for patients with epilepsy, and whether there should be safeguards to ensure both physician and patients are informed when generic substitution occurs. It is the substitution of a prescribed branded drug by different products of the same active substance. The generic is usually unbranded, and it is bioequivalent to the brand product. Generic substitution becomes possible when the patent life of the active substance has expired. And here, what we mean by bioequivalence is the property of two dosage forms or active ingredients with the similar blood concentration levels that produce the same effect at the site of physiologic activity. When we talk statistically about this, the criteria set by the majority of the regulatory authorities of two products to be bioequivalent is that the area under the curve and the Cmax, or maximum concentration ratios of both products, they mean the brand and the generic, should fall within the range of 80 to 125%. This is the 90% confidence interval. So as we see here, that the upper limit is 125% and the lower limit is 80%. And also we have the definition of pharmaceutical equivalent. So both products should have the same active ingredient, and they are of the same dosage form and route of administration, and they are identical in strength or concentration. So we have the definition of bioequivalence and the definition of pharmaceutical equivalence. Actually, they both add up to give us the definition of therapeutic equivalence. Also, although many of the papers are talking about the bioequivalence as the main property to define the generic to be allowed to be substituted by the brand, is to be bioequivalent only, not to be a pharmaceutical equivalent. So the best method to ensure the therapeutic equivalency of pharmaceutical products is the bioequivalence, and the bioequivalency of generic products have been approved by the FDA since the enforcement of the Drug Price Competition and Patent Term Registration Act. This was in 1984. According to the FDA, when two drugs are bioequivalent, it means that both of them will provide similar and desired clinical effects. In some markets, like in UK, generic products can only be dispensed against prescriptions that are written generically. Substitution of generic products for a prescription written as a brand name is not permitted. But in other markets, like in Canada, generic substitution, like dispensing of a generic version, if one is available, even if the physician used the brand name on the prescription, it is allowed or mandated. If the physician don't want if the physician doesn't want to substitute the generic product, then he has to indicate this on the prescription or by filling in other exemption form. Although healthcare systems tend to favor the use of generic products because of the savings in cost of medicine, there are a number of potential disadvantages to the use of generic products. In majority of therapeutic areas, these disadvantages are irrelevant or minor in comparison to the economic benefits. However, many healthcare systems recognize that changing the supplier of medicines could compromise care of patients with certain conditions. Physicians are advised to prescribe these products by brand name, and such products are exempt from generic substitution. A number of experts and professional bodies have recommended caution when generic substitution of drug requires for epilepsy. Many antiepileptic drugs are considered to be treatment with a narrow therapeutic index, only small relevant differences in dose between the therapeutic and the toxic effect. So a narrow therapeutic index implies that slight variation in the drug absorption could result in significant negative health outcome. The FDA considers drug to have a narrow therapeutic index if it has less than twofold difference between the minimum toxic concentration and the minimum effective concentration, and safe, and it has to be safe and effective use requires careful titration and patient monitoring. Here are some of the factors increasing likelihood of problems with generic substitution in some of the antiepileptic medication, like phenytoin, carbamazepine, and valproate. We can see that phenytoin has low water solubility and narrow therapeutic index and nonlinear pharmacokinetics. Carbamazepine has also low water solubility and narrow therapeutic index. Valproate has narrow therapeutic index and nonlinear pharmacokinetics. So with all antiepileptic drugs, there is wide variation in therapeutic dose among individuals, and to ensure that the optimal dose of antiepileptic drug is used and to avoid the toxicity, dosage is usually adjusted over a considerable period of time, according to the response of the individual. Even antiepileptic drugs with low toxicity and with a wide therapeutic index, such as lamotrigine, require titration to the optimal dose. If adequate control of seizure is not achieved with a tolerable dose of one or two antiepileptic drugs in sequential monotherapy, then other antiepileptic drugs are often added to as adjunctive therapy, again with titration according to the therapeutic response. As a result, Many patients with epilepsy are on a daily regimen of multiple treatments that has been carefully adjusted to obtain the optimal response. Many antiepileptic drugs induce hepatic picrosomal enzyme, increasing the rate of metabolism of concomitant drugs, and this is further complicates the dosage of antiepileptic drugs in patients receiving polypharmacy, adding to concerns about the potential for adverse consequences if aspect of this finally balanced therapy is disturbed. So as a result, there is a considerable reluctance from physicians and patients to change the therapeutic agent in some patients, particularly once stability has been achieved. So in some clinical experience that they examined the use of generic products. One study of the patients treated with carbamazepine, valproate, and phenytoin found that 18.7% of respondents experienced a switch in medication supplier in their previous 2 years, and of these 10.8% received problems after switching that were validated by their general practitioner. And one study initiated by the FDA comparing carbamazepine brand to 3 generic formulations, they found that these formulations were absorbed significantly more rapidly than the innovator brand. However, the author concluded that these differences would unlikely to have clinical effect, and the generic formulations were comparable to each other. In an open study, 64 patients with seizure disorder and mental retardation were randomly assigned to generic valproate or continued to receive branded innovative product Tipicaine for 4 weeks, and were then switched to the other therapy. There were no significant changes in blood levels of the products or in seizure frequency, though the same manufacturer produced the capsule for both of the branded and the generic. The same manufacturer is producing the capsule for both brand and the generic. The author concluded that switching from the brand product, which was $94 for the box of 100 capsules, to the generic product, which was $7, would result in a considerable cost saving. Okay, here's the again just a revision about the pharmacokinetics characters of the antiepileptic drugs, which may increase the probability of problems associated with the generic substitution, like the narrow therapeutic index, the low water solubility, and the non-linear pharmacokinetics, and also hepatic enzyme induction or inhibition. If we want to say briefly about the advantages of generic substitution, of course, the first thing is the lower cost. The generic drugs does not have to undergo the large, expensive clinical trials that are required for the approval of brand drug. Also, the compliance will be increased. It will be better with generic compared to their brand name equivalent, with lower co-payment being given as a key factor. Also, the quality generic drugs are produced under strict standards to guarantee their quality. What are the disadvantages? Increase the breakthrough seizure with generic substitution, failure of drug therapy with generic substitution, and toxicity, and increase the serum levels with generic substitution. Here again, the list of antiepileptic medications with their pharmacokinetics parameters that we see, and most of them would have 1 or 2 of the pharmacokinetic properties that would interfere with the visibility of changing from switching from generic to the from brand to the generic. So what was the recommendations of the American Academy of Neurology about the generic substitution? They mentioned that generic substitution can be approved only if safety and efficacy are not compromised, and patients should avoid switching between formulations of antiepileptic drugs. Physicians should avoid switching between formulations of antiepileptic drugs. Specific information about each antiepileptic drug, like the area under the curve and Cmax reported complications, should be made available to the physicians, and the pharmacist should be required to inform patients and physicians when switching a patient between manufacturers, with labeling to allow identification of specific manufacturers. And organizations that encourages or mandates the substitution of antiepileptic drugs, like federal or state agencies, should evaluate their responsibility for problems arising from this policy, and further research on the impact of generic substitution is required. That was all, and I hope that it would be a fast coverage, like introduction for the topic. Thank you.
Okay, thank you very much, Dr. Lina, for your nice introduction. That was a nice overview about the topic. So as we mentioned, we're going to leave all the questions and the comment to the last part of the webinar. So here, if I can kindly introduce Dr. Yusuf bin Jardan, who is a very brilliant pharmacist. He is assistant professor in College of Pharmacy in King Saud University, and he is also the leading in charge for the animal lab in the King Saud University Pharmacy College. So he is so he's going to talk about the technicalities of the process of how we comparing and how we approving the various branding for antiepileptic medications. He's going to having an overview about the topic, and so Dr. Yusuf, it's our pleasure to have you on board with the Saudi Epilepsy Society webinar series of medications. So Dr. Yusuf, I can still see your slide. Dr. Yusuf, if you can unmute yourself.
Yes. Sorry about that. So thank you, Dr. Majid, for the kind invitation for this topic. And I would like thank everybody for joining us tonight. So today I will cover the bioavailability as the important pharmacokinetics parameter. So in this topic, I will be okay. So what do you expect from this topic, from this sessions or from this lecture? So to understand different terms like pharmacokinetics related to the parameters, also to know the importance of bioavailability in pharmaceutical industry. So also to consider the changes in bioavailability in clinical settings and to understand the methods of assessing the bioavailability. So for this short topics, I will cover the bioavailability, what's the bioavailability, the importance of the bioavailability, also how do you assess the bioavailability in the lab settings or in the regulatory certain situations, also to method the assessing the bioavailability. Also will compare and to know about the relative bioavailability and the absolute bioavailability terms. So at the beginning, what's the bioavailability? There are different definitions, and some definitions are similar to each other. So we can say the bioavailability can be defined by a measure of the rate and the amount of drug which lead the systemic circulation and change following the administration of the dosage form. Also, if there is a simple way to explain or to define the bioavailability, it's the fraction of administered dose that actually reach the systemic circulation. If you see in my slides, I will talk a little from academia point of view. So the bioavailability is important, very important in developing a suitable dosage for a new drug. Also, it will defining the influence of excipient, its possible interaction with the dose and the efficacy on the absorption. Also, it might improving the existing dosage form. Also, it will be assessing the both safety and the efficacy of the drug product. So usually for the assessment of bioavailability, as you know, when we start with a new drug or a new medication, we start at the preclinical stage when we have in vivo studies on rats or dogs or monkeys. Then after that, we will be, if we're having a similar data in different species, so in this case, we'll estimate that bioavailability in the human in general. So there are different requirements for bioavailability studies. If you have a not marketed drug, which is called the full in the new drug application, so we have to record this data for this drug. Also, if we have a new formulation of the active drug ingredients or therapeutic moieties that have a full NDA approval or the approved for marketing, so any changes of the excipients of the dosage form, it might have a change on the bioavailability value of this medication. So for the drug with the NDA, so we need to characterize the essential pharmacokinetics parameter, which is the rate and the extent of systemic absorption. Also, we'll estimate the elimination half-life and the rate of excretions, also metabolism after a single or multiple dosage administration. So for the bioavailability, there are like different factors. It might affecting the bioavailability of the drug. Usually, the physicochemical properties of the drug, its solubility, like the pKa value and other values. Also, if like physiological factor, it might be changing the bioavailability in this case. So, like we might have like a healthy patient which have the comparable bioavailability value to the diseased patient. Also, the type of dosage form. If we have like a solution, it will have a different bioavailability value compared to the tablet or the capsule dosage form. So, but for the like new formulations of the drug, so we have to characterize the pharmacokinetics of any new formulation, new dosage form, any new salt or ester related to the reference. So in this case, we have to conduct a full bioequivalence studies. So for the important part for the assessing bioavailability, we have like different methods to assess the bioavailability. The first one, which is called the acute pharmacological effect, and the second one is called the plasma drug concentration. Also, we'll have the urinary drug excretions method, and also we'll have the like comparative clinical trials or the in vitro measurement of bioequivalence, which is like kind of in vitro study for the solution testing. So for the acute pharmacological effect, usually we will like see on any changes to the pharmacodynamics parameters. So it's used for there is no assay for the drug plasma drug concentration. So if we have the if we don't have a valid sensitive method, so we can use this method as the assessment for the bioavailability. Also, if we use if the plasma concentration may not reflect the bioavailability of the drug at the side of actions for usually like locally active or locally acting drugs. Also, we might be quantifications of the pharmacological effect versus the time by using a measure of the blood pressure or the heart rate or the convulsions and the others. So for the acute pharmacological aspect, so from this data, we can consider in this fact the onset time, intensity and the duration of action, also the therapeutic window of the of the drug. So as you see in this figure, we have different pharmacological and pharmacodynamic and pharmacokinetics parameters. So this is like a hypothetical like figure of the plasma versus time after a given drug as a single dose. Let's see orally. So if we have different parameters here, as you see, if we give the drug, the drug goes up until the Cmax, then it will go down until the post-absorption phase, and we'll hold also we have the elimination phase at the end. If you see, we have different pharmacodynamic or pharmacological factors, or the parameters is called the onset of action, and if you see the duration of actions, these are kind of pharmacological way to assess the bioavailability. And for the pharmacokinetics parameter for the plasma drug concentrations, we have a very important parameter, which is the Cmax, Tmax as well, and also we have the AUC. Will tell us the measure of the extent of the drug absorption. So the important parameters, we can start with the Tmax, which is the time of peak plasma concentration. So there are different definitions and different references or different like regulatory recommendations. It's like simply we can define this parameter as time required to achieve the peak concentration of the drug after dosing as the indication of the rate of the absorption. Also, we have the Cmax, which is the peak plasma drug concentration, which is the maximum concentration of the drug obtained after the dosing of a single dose of the drug. And another very important parameter, which is the AUC, which is called the area under the plasma concentration time pair. So there are different ways to measure this one, but the simplest way or the widely used, which is called the trapezoidal rule, is the measurement of the extent of the drug bioavailability. So if we can say the AUC reflects the total amount of the active drug that reached the systemic circulation, and it's the most reliable measure of the drug's bioavailability. So usually AUC is directly proportional to the total amount of the unchanged drug which reached the systemic circulation. So the third one, which is the urinary drug excretion. So it's most accurate if we have the active therapeutic moiety which is excreted in significant quantity as unchanged in the urine. urinary excretion of the anti-drug is directly proportional to the plasma concentration if it's like at 10% or 20% excreted. So this method is like non-invasive, which is can be easy to do it if we have a drug, it's excreted as unchanged in the urine. So so if you have this figure, there is a correlation between the plasma drug concentration and with the cumulative drug excretion. As you see in the, like if we are given a drug, if you see like in plasma in the blue line here, if you see if you're given a dose, it will goes down. This is like IV dosing. So if it goes down until to be like reach the like lower concentration at 24 hours. If you see the red line here, we have the data from the urinary excretions. As you see, when the time goes up, if you have something from 0 to 20, if you have the drug excreted in the urine, it can't be measured. So we have different parameters here. It's very important to know the time for the maximum excretion rate, and also we have the cumulative amount that excreted in the urine as it increases, as the extent of absorption increases. Also, we have the maximum urinary excretion rate, which the value increases as the rate and the extent of absorption increase. So this was obtained from the plot of the peak plot between the rate of excretion versus the midpoints of time of the urine collection period. So we can conclude from these two important methods, the plasma concentrations method and the urinary, we have different parameters. We have to focus on them. They are the Tmax, Cmax, and AUC for the plasma data. And we have different parameters to focus on in the urine data, which is the T infinity max, and we have also the cumulative amount of drug excreted, and also the maximum urinary excretion rate. So also we have the fourth like methods with the comparative clinical trials. It's used to measure bioavailability quantitatively. So it's less precise because of the inter-subject differences. It might be like this, we can see variable in this kind of studies. And also we have the in vitro measurement, which is the correlate between the rate of the drug dissolution in vitro with the drug bioavailability in vivo. So in this methods, we are having the experiments in the different dissolutions methods according to different pharmacopoeia, which are the United States pharmacopoeia or the like British pharmacopoeia. So now we'll talk about a very important parameter, or if you have the data from the bioavailability study, we have the relative bioavailability and we have the absolute bioavailability. Let's say for the we are just having the systemic bioavailability of the drug from the dosage form is compared to a reference standard given by the same route of administration. So by having the data, the plasma concentrations data for both formulations or for both generic or brand, so we can estimate the bioavailability value for this by using different equations, which is the one of the important one, which is the AUC of the product A by the divided by the AUC of the product B times the dose of the B divided over the dose of the product A. The second one, which is the absolute bioavailability, it's the comparing the bioavailability, which is estimating as the drug under the care or IUC of the active drug in systemic circulation following intravenous administration after the oral or the rectal or the transdermal administration with the bioavailability of the same drug following the intravenous administration. If you see, we have different. You have the IV dosing, which is the orange color here, and we have also if you can see, this is the oral one. So we are comparing the bioavailability value by having the AUC value for both the IV and the oral, also the dose and the oral the oral dose of the oral dosage form times 100 will have the estimation of the bioavailability, the absolute bioavailability. So for the summary of this lectures, we can see different between the bioavailability and bioequivalence in terms of how to estimate them. And bioavailability is an important parameter to consider in choosing the drug therapy. Also, bioavailability is the key pharmacokinetics parameter which must be systematically estimated for new drug or formulation. Any changes in the bioavailability, it might has change in the other parameters. So monitoring the plasma concentration is a very important factor. If you have generic or brand, you have to having these parameters in your account. Also, having a laboratory equipment with certain ethical methods, which they are sensitive and selective, it will help you in estimating this parameter. So switching from different brand or generic as well as certain dosage form needs special care in this case. So you have to consider, do we have the AUC value similar compared to of the brand compared to the generic or not? What the bioavailability rate of or the value for the brand or for the generic? And it's very important to note that most of the regulatory bodies here in Saudi Arabia, on the United States or different countries, they are asking all the manufacturing company to submit all this data or all this information to them to assist them and also to give the approval for the drug to be in the market. Thank you so much for listening. I hope you enjoy the simple way of the lecture.
Okay, thank you very much, Dr. Yusuf, for the nice overview for a very complicated topic. So as we mentioned before, for the audience, I mean, if you have any questions, you can submit it through the chat, and we're gonna answer all the questions at the end of the talk. Now we're gonna shift gear from more of a technical aspect of the topic to more of a clinical view or light on this topic. So here we're going to introduce our guest speakers, Dr. Emily Johnson. She's an assistant professor in Johns Hopkins Epilepsy Centers. She focuses on the treatment and diagnosis of epilepsies. She has a clinical interest in the women with childbearing age with epilepsy. Her interest also including the bioequivalence for the antiepileptic medication. She has many publications in this regard, and we're very delighted to have her here to share her experience, view of the topic to all the audience here in Saudi Arabia. So Dr. Johnson, the podium is yours.
Wonderful. Thank you so much for the kind invitation and for this opportunity to talk about this important topic. And thank you for those really great talks on bioequivalence. So I'll touch briefly on bioequivalence, but you've already heard it covered in much more detail by these experts. I'll talk about clinical observations in studies from brand to generic switching, and also the topic of generic to generic switching. I'll touch on the special topic of modified release antiepileptic drugs, which are a special challenge. And then I'll also talk about patient experiences and patient examples. So first I'll talk about what is bioequivalence according to the FDA. So the way, as you heard, the way that the FDA approves generic drugs or generic formulations is by looking at single dose crossover pharmacokinetic studies in healthy volunteers. And these can be a fairly homogeneous population. Sometimes it's all men in the group. They might be all from one country. And they look at the area under the curve, and they look at the Cmax, the peak concentration. And they look at this under both fasting and fed conditions. And the 90% confidence interval for those two conditions has to be within 80% to 125% of the reference, that is of the brand name. So that is a fairly wide range. Of note, they don't take the Tmax, or the time to the maximum concentration, into their decision-making process when they approve a new generic. So for example, if this blue box is the reference drug or the brand drug, then of these three generics, A, B, and C, A and B could both get approved, even though B has a very wide confidence interval, because they both fall within that 80 to 125% bound. But drug C, even though it has a narrower confidence interval, the confidence interval crosses 125% of the brand, and so it would not be approved. So this is a comparison of how closely some generic and reference products match in terms of the area under the curve. So this is looking at different medications here along the bottom. You can see on the left, each one is tested in the fasting state, and on the right is each one in the fed state. The green represents those with a smaller difference, and then the red is a larger difference. So you can see that some of the drugs like levetiracetam have a very, very narrow, or have a very close approximations in terms of AUC, both in the fed and the fasting condition. But you can see for some of the other drugs like gabapentin, there's a lot more that have a larger difference. This is the same type of representation, but now for Cmax instead of AUC. And you can see that more of the drugs have this red, so have a larger, greater than 15 to 25% difference between the generic and the brand product. And so even levetiracetam, which was the easiest to copy in terms of AUC, has more differences in terms of the Cmax. So this just shows that not all products are the same in terms of having generics replicate the brand. All right, let's talk about brand generic switching and some studies that have shown problems when this has been done. So there have been a number of case series reporting patients having seizures after being switched from a brand to a generic product. This was a study from Minnesota in the US looking at a series of patients who had been well controlled on Dilantin, and then they suddenly had breakthrough seizures. So they examined them and they found that they had 11 patients who had been switched from brand to generic. And of those, 8 patients had breakthrough seizures. And so they tested these patients. They tested the total and the unbound phenytoin serum concentration. And they found that when they were getting the brand name, they had a higher concentration that then decreased when they were given the generic. They then switched them back to the brand and they found that this led to a higher level. So this is an anecdotal case series, but highlighting that there can be problems or there can be differences in concentrations when people are switched to a generic. So people have tried to study this important issue in a number of ways. One way is by looking at switchbacks. So in Canada, as we heard earlier, when a generic product comes out on the market, most patients are automatically switched to a generic. So this was an examination of pharmacy claims in Ontario, Canada. And they looked at once a generic came in the market, they looked at the number of patients that were switched to the generic, and then how many of those were switched back to the brand. They found, so they compared depakine and Lamictal to 3 different non-antiepileptic drugs, to a statin and to 2 SSRIs. And they found that the rates of switchbacks, so that is, they were on the brand, they were switched to the generic, and then they switched back. The rate of switchbacks was much higher on AEDs than on other types of medications. It was between 12 and 20% on AEDs, whereas it was much lower, about 1 to 2% on other types of medications. They also found that of those patients who stayed on generic lamotrigine, many of them actually then had a dose increase or had an addition of another anti-epileptic drug. So they were, so this is evidence that there can be problems, there can be reasons for people to switch back or to increase therapy after being switched from a brand to a generic. This is a similar examination, but now in Quebec. And they again found that patients who were switched from brand to generic AEDs had more frequent switchbacks to brand than did other types of drugs. In this study, between 20 to 44% switched back from a generic to a brand, whereas in the other medications that they looked at, that was carvedilol, fascinepril, and simvastatin, the switchback rate was much lower at only 7 to 9%. However, people have pointed out that there's a lot of problems with looking at switchbacks as a measure for seizure frequency. You can imagine if you have a patient who's having seizures every 3 months or so, they'll probably continue to have that seizure pattern with a seizure every 3 months, unless you increase their medication or unless they have surgery or unless something else happens. So you can imagine a patient who is destined to have seizures every 3 months. If they got switched, if they were on the brand name drug and they were having seizures, and then 2 months after their last seizure, if they got switched to a generic medication, they then had a seizure with their typical 3-month interval, that could be concerning because they could look back and say, oh, I just changed to a generic medication. I wonder if that caused my seizure. And you can foresee that some physicians and some patients would want to switch back to a brand at that point. But it's hard to distinguish, would that person have had a seizure anyway because it was, they would have had it even had they remained on the brand, or was it due to the brand generic switch? So that's one problem with these switchback studies. Another method of looking at this, of trying to determine how clinically important it is to look at brand generic switching and then hospitalization or medical usage afterwards. So this was a study looking at 400 people with epilepsy who had had hospitalizations or who had had emergency room visits. And they looked, for each patient, they then looked at 3 controls. And they found that those who were cases, that is those who had used medical resources, were more likely to have had a formulation switch in the past 4 months prior to that hospitalization than were the controls. And so it was relatively low rates. They found that 3% of people had had seizures within had had a switch within the previous month prior to the hospitalization. But they did see that elevated rate even up to 4 months prior to the hospitalization. These, and of note, these were any kind of formulation switch. So it could have been brand to generic, it could have been generic to generic, or it could have been generic to brand. So this is some additional evidence that people can have breakthrough seizures after being switched from brand to generic or in a different direction. Okay, but these are retrospective studies. So people have done prospective studies as well, where they look at people who are going to be switched to another formulation. So this was one prospective study that came out recently in Poland. That for some reason, the price of brand trileptal was going to increase by tenfold. I'm not sure why. So many patients who were on trileptal were going to switch to generic oxcarbazepine. So they looked at 103 patients. Of those, 27 stayed on the brand, even though it increased in price, but 76 switched to generic. And in those patients who switched, they did not find any increase in seizure rates or increase in adverse effects. So that's a prospective study, smaller numbers than we were talking about before. Here's another prospective study that came out in 2018, looking at patients on Keppra who were going to switch to generic. They found that of those 148 patients, 109 had been seizure-free prior to the switch. And they found that 105 of those 109 remained seizure-free. Of all the patients together, they did see increased seizures in 7, which was about 5%. However, they found that 10 patients actually had decreased seizures, which was about 7%. So that makes it difficult to say, was switching from brand to generic really the cause? Were people going to have seizures anyway? Was it just their time? Or did some people actually benefit from that? But I do want to point out that the number of people reporting increased seizures, about 5%, is relatively low. This is a systematic review looking at 20 articles. And interestingly, they looked at both retrospective and prospective studies. And they found that retrospective studies all did show a significant relationship between generic substitution and increased use of healthcare resources, which they use as a proxy for seizures. So that was retrospective studies. However, looking at prospective studies, they found that 6 prospective studies in epilepsy patients actually had no differences in the PK parameters or in seizure frequency after patients were switched. They also found 7 prospective studies in healthy adults, which did show some differences in PK parameters. So we're seeing a discrepancy here between retrospective studies and between prospective studies. So we've been talking about brand generic switching. Let's also talk about generic switching. So one problem that can happen when people are switched between different generic manufacturers is the pill appearance can change drastically. So generics made by different manufacturers can have a different color, size, or even shape. Some studies of other types of medications have found that the chance of a patient having non-adherence or non-compliance increased by 34% after just a change in color, but up to 66% after a change in shape. This was not data specifically from epilepsy medications. This is data from cardiac medications. So if you are used to getting your yellow oval pills for your levetiracetam, and then all of a sudden you're switched to a slightly different shape, white pills, you might be confused, you might forget to take them, you might not know that you're supposed to keep taking them. So they did find that this significantly affects patient compliance and adherence. So these are just 2 different versions of levetiracetam 500 milligrams that I found. Another difference is inactive ingredients. So when you switch manufacturers, generic manufacturers may use different inactive ingredients as the binding agent or the filling agent. And some patients may have a sensitivity not to the active ingredient itself, but to a binding agent or to one of the inactive ingredients. All right, also there's a concern between disparate generics. So as we talked about before, these drugs need to be approved by having confidence intervals that fall within the 80 to 125% confidence interval of the brand name drug. However, that leaves a lot of room. There are cases where one generic has levels much closer to the 80% range, and one generic has levels much closer to the 125% range. So there's a big concern that if you switch from a high generic to a low generic, this could actually significantly affect the drug level in the patient's blood. So if you are this is an example from our earlier figure. Again, the brand is blue and 2 different generics at each end of the approvable spectrum. are in green there. If you switched from the high to the low, you could anticipate that there might be a problem. So the US FDA was very concerned about this and wanted to do a study to see whether that makes a clinical difference. So this was called the Equigen study. It stands for the equivalence of generic drugs. And this was a study of lamotrigine. And you can say that there were two arms to this study. And they took a generic that was on the high end of the range and a generic that was on the low end of the range for lamotrigine. And they had patients switch back and forth between the low and the high. So patients were randomized after having an observed baseline to either start with the high generic for 14 days, then go to the low generic for 14 days, again switch to the high, and again switch to the low. So this was an 8-week study. Or they could have started with the low and switched to the high. And so they were evaluated every 14 days, and they had PK parameters drawn, and they had seizure diaries recorded. So this was a very thorough study of switching between high and low generics. It was a small study. There were only 33 patients, but they had identified these people as patients that they suspected would be generic brittle. And so when we think about generic brittleness, we think about patients who have reported seizures before in the setting of switching from brand to generic, or patients who just have a high likelihood of having breakthrough seizures. So they chose 33 generic brittle patients, and they switched them between high and low generic lamotrigine in 4 segments. So they changed them over 4 times. They found that when they examined these PK parameters, they actually found that the 90% confidence intervals did indeed fall within these tight limits of the brand name. And they actually found no significant changes in seizure frequency or in adverse effects, even when people were switching between the high and the low generic. So that was good evidence that although we can measure some differences, it may not make that much clinical difference. So let's talk about modified release AEDs, or long-acting, extended release, a lot of different names for them. So modified release AEDs are a unique case because there are multiple different release technologies that can be used to make a drug modified release. There can be a solid matrix that dissolves in the gastrointestinal system. There can be an osmotic pump where there's pores, and then the drug slowly diffuses out of the pores as it equalizes with GI fluids. Or there can be coated microgranules, which release at different times. Of these modified release AEDs, some are still BID formulations, such as carbamazepine, but many are daily formulations. So they have to try to get much more even 24-hour coverage by using some of these release technologies. And each product may actually use more than one release technology, and that's actually proprietary information. So Carbitrol, the brand, uses 2 different technologies. Tegretol XR uses 2 different technologies. You can see Keppra XR actually uses 3 different technologies. And it's proprietary, so there's actually a column for unknown technologies because the companies are not required to disclose their information. So these are some pharmacokinetic profiles from modified release anti-epileptic drugs that we requested from the FDA. And each of these, number 1 represents the test or the generic that's under approval, and number 2 represents the brand or the reference product. So you can see this is for levetiracetam 500 milligrams in the fasting state. So you can see these are very, very close. This is a really good copy in this generic formulation. Here, I think this is very interesting. This is the PK profile for modified release divalproex. I think this is interesting because you see in each, the brand and the generic, there are 2 peaks there, probably reflecting the absorption with 2 different modified release technologies. But you can see that the generic, the solid circles there, did quite a good job of copying the pharmacokinetic profile of divalproex. But you can see this is a carbamazepine, 400 milligrams modified release. You can see that the Cmax might be somewhat different. It was still within that 90% confidence interval for it to be approved, but you can see it was a bit different. And here's one. This is lamotrigine 100 milligrams fasting. The brand is the open squares, and the generic is the solid circles. You can see that here, while the AUC and the Cmax are fairly similar, the Tmax is quite different. The Tmax for the generic was about 10 hours, where the Tmax for the Tmax for the brand was much longer, was more along the lines of nearly 30 hours. So you can see that this could have a clinically significant implication for a patient if they're sensitive to side effects at the time when they hit their Cmax. So if their Tmax is occurring while they're awake versus when they're asleep, you can see that that might make a clinically significant difference in their quality of life. All right, this is a study that I did with Greg Krause, a colleague here. We looked at intersubject variability in the AUC and the Cmax, and we compared that intersubject variability between immediate release and modified release antiepileptics. And we found that while the intersubject variability was fairly similar between generic and reference drugs, modified AEDs actually had higher intersubject variability than did immediate release AEDs, especially for lamotrigine. That's had the most intersubject variability. We also looked at differences between fed and fasted conditions. And we found that while the AUC and the Cmax also had differences, the Tmax had significant differences, where many products had a difference of 20% or greater when you looked at the fed condition versus the fasting condition. Interestingly, it wasn't in the same direction for all products. Some products had a higher or had a larger difference in the fed condition, and some had a larger difference in the fasting condition. All right, so let's talk. So we've looked at a lot of studies and a lot of data from bioequivalent studies. Let's look at some patient perspectives. So these are some quotations from the Epilepsy Foundation website. There's many more in there. The first one says, our daughter switched to the generic Zonogram and within 1 month was having numerous breakthrough seizures. We switched her back and the breakthrough seizures went back to their usual number and regularity. It's a family in North Carolina. And another one talks about, my daughter was recently put on generic Trileptal and experienced really bad side effects. We had to take her to the ER twice within 1 week because of severe ataxia. She was unable to walk, sit up, or eat. After running many blood tests, it was believed that it was the generic Trileptal. Once we switched her back to the generic name, or excuse me, to the brand name Trileptal, her condition improved. Her neurologist stated that he had had other patients experiencing similar side effects. So this is a big concern, and people do notice some differences sometimes when they are switched. This is a report of another Epilepsy Foundation survey. This was a survey that was on their website between 2006 and 2009, and we had over 1,000 respondents. However, just a note of caution, these are people who were self-selecting, which means they were probably more likely to be experiencing problems, which is why they sought out this information and took this survey. So people were asked about breakthrough seizures and having AED switching. Almost 70% had switched from brand to generic. In this very self-selecting group, 60% reported worsening seizure control and 50% reported worsening side effects. Interestingly, another 40% had had a brand to generic switch. And of those, 15% had worsening seizures and 18% had worsening side effects. So you can report problems going the other direction as well. But again, keep in mind, this is a very self-selecting group of patients who were interested in this topic, who sought it out, and who visited this website and took this survey. Here's another issue that sometimes comes up. I talked about different modified release technologies. So we've had patients who are switched from brand name to Teva Pharmaceuticals, generic levetiracetam, or between generics, and they ended up on Teva Pharmaceuticals. And we've had multiple patients report ghost capsules that they found in their stool. And so they're very concerned that that means that the medication didn't absorb and that the medication is not being effective because it's passing through them unchanged. So actually, there was some investigation. Later tests revealed that these are truly empty shells. It's a special enteric, it's a special osmotic pump releasing formulation, and that all the levetiracetam active ingredient has actually already been dispensed, but the shells pass through unchanged. But this is really concerning to patients, especially if they've just switched formulations and they haven't noticed it before. They're very worried that they're going to have breakthrough seizures. I've had a number of patients who report sensitivities, which we think is due to an inactive ingredient. I have one patient who had very dry eyes on one formulation of generic lamotrigine, but that improved when he went back to his former generic manufacturer. I've had people with itching and rash on one type of generic levetiracetam that improves when they go back to another generic. And I've had people with hallucinations on one formulation of generic levetiracetam that improved on another formulation. So people can have these idiosyncratic sensitivities to certain generic manufacturers. When switching from a brand to a generic, many patients are so concerned that they request to be off work. I had a lovely woman who was a teacher. She had been on brand name Lamictal for many years. She was seizure free. but it was too expensive for her. She wanted to switch to generic lamotrigine. And she actually wanted to be off work because she was so afraid of having a seizure in her classroom. We ended up doing the switch during the summer, and she was completely fine, but it was just such a source of anxiety for her that she didn't think she could work during it. Many patients consider this, they consider the switch for months before switching. I recently had a young woman who had been on brand named Tegretol for 14 years. She was doing very well. She'd had a surgery and she was actually seizure-free for many years. And she had been discussing switching from brand name Tegretol to generic for about 6 months before she was ready to do it. So it's a source of high anxiety. It's a time of high anxiety. And it's a real concern for many patients. Others ask about, do they need supervision during this time in case they have a seizure, or should they stop driving? I have a patient who was seizure-free, but she needed to switch from a brand to generic. She was so worried that she ended up getting a seizure detection device. She got the Embrace watch because she thought that it would give her more confidence that if she had a seizure, someone would be able to be notified. So it's a really important and serious concern for many patients. So this is my approach. I do start patients on a generic drug when there's one available. If there's a generic available, my approach is not to start someone on brand. And I request that the pharmacy maintains the same generic manufacturer if there's problems with switching. And so most of our pharmacies are very willing to work with us and to work with the patient to get the same generic manufacturer, especially if they've had a problem before. If a patient has always been on brand, I will help them switch to a generic if they desire a more cost-effective option. I do check a level before switching and then after switching. At this point, I don't have them stop driving because I think it's, for most patients, it's such a low likelihood of having a problem. And I will request brand only if the patient has had breakthrough seizures in the past on generics. In my experience, this has been quite a small number of patients, but as you saw from some of these earlier studies, it's not zero. It's certainly a concern for many people. So that's just my approach. I do try to start people on generics when possible. I think this will come up soon in the next year or so once Vimpat becomes generic, because I have many patients on Vimpat who it's an effective medication for, but they don't like the price. So I think this will become very relevant soon as we're dealing with the generics coming on the market and people want to switch. Perfect. Well, thank you very much for this opportunity. There's my, I'll stop sharing there.
Okay, thank you very much, Dr. Johnson, for this very brilliant highlight for the topic. As we can see from all the speakers, it's very not an easy topic to digest from one aspect, and it's more challenging when it comes from the patient perspectives. So may I kindly ask all the speakers to unmute themselves in case of any questions comes. So I have a few questions come from the speakers, for the speakers and the audience. And one question, first of all, he thanks for all the speakers for the impressive presentations. He's asking, is the manufacturers of the generic anti-seizure medication is the one doing the bioequivalence investigations and providing the data to the regulatory authorities, or the regulatory authorities are doing it by themselves? So, Dr. Johnson, do you have any questions about that? Or?
Yeah, in the United States, it is the companies themselves who are doing the studies and then are presenting the data to the FDA. Yeah, in all the cases that I'm aware of, it's the generic manufacturers are responsible for doing the studies. Okay. So here is another question, maybe Dr. Lina. There are questions from a family physician practicing here. He's saying that at one point he was asked to write the brand name or the scientific name for the drugs only, not to mention any brand name or any specific names. And that should be left for the pharmacist to dispense the medicine as it sees available in its own stock. So what do you think about that? Dr.
Can you repeat the question? I didn't get the idea of the question.
Yeah, so he was asked not to specify any names for the medicine, for various generic names. He just asked the the generic name or this the scientific version of the name of the medicine, and then the pharmacy will dispense the medicine as he or she available in his her store.
Yeah, so this is what they are following in their hospital. I mean, every institution, there should be a policy regarding the automatic substitution, they call it. So if this is applies to the institution where she's working in, when she received any, maybe like in our hospital, when we receive a prescription, it is mandated from the policy that the physician write the prescription by the generic name, not by the brand name. And then it's up to the, the pharmacy, the hospital will have their stock or the formulary that they will bring the medication from certain company according to the budget they have annually. And then the pharmacist will dispense the product that they have in hand in their in the stock. So it ups to the institution policy.
Okay. So like in your personal opinion, what do you think about that?
Of course, all the companies that are allowed to be they have a contract with the hospital to get the stock from, they have their own evidence for their bioequivalence studies that they provided to the authorities like the Saudi FDA before they are allowed to sell or they market their products. So I think this is going to be have a good impact on the hospital's budget, and it will open the floor for the competencies between the companies to produce good quality products with lower price.
Okay, thank you. This is also questions we get from the audience. He mentioned that epilepsy is an episodic conditions, and this makes more clear to a difference between drugs' clinical effect. The major problems comes to other healthcare issues like hypertension, where use generic, less effective than medications, and the titration, and that could lead to severe complication like intracerebral bleed. I would say this is just beyond anti-seizure medications, but this is more of a general concept for disease and related to switching brands versus generic. So I don't know, Dr. Yusuf or Dr. Emily, if you want to take the question.
That's an issue. Oh, go ahead.
Yeah, go ahead, Dr. Emily. I didn't have anything to add on this topic. It's out of my scope, so.
It's an interesting point. You can make an argument that in epilepsy, it's actually harder to know soon. You don't know if there's a problem until the patient has a seizure. Whereas if it was an antihypertensive, you could foresee carefully checking the blood pressure after they switch to the generic and hopefully intervening before there was a severe problem. But that's a really interesting point about the continuous versus episodic disease.
Okay, so Dr. Yusuf, there is another question saying that the neurotherapeutic index, how accurately this can be measured in the bioequivalence trial?
Like it's they have like the same criteria as we mentioned in the lecture, but they have some specific like parameters they have to keep in account, and usually it's required by the regulatory bodies. As here in the Saudi FDA, they have their certain guidelines, certain policy for different kinds of medications.
Okay, thank you. Here's another questions from one of the audience. He's asking you specifically about the clobazam. He's saying that we're facing major problems in clobazam, where there are big differences in clinical effects if we move from drug to drug by different manufacturers. Despite this clear clinical non-equivalence, still we do have these problems. I think this is based on his clinical practice that clobazam there is a variabilities by different drugs. So maybe Dr. Johnson, if you can share a point on this.
Yeah, that's really interesting. There's some thought that some drugs are much more prone to having generic problems. For a while, I think it was lamotrigine was a drug prone to having generic brittle problems, but clobazam I'm not surprised to hear. I think we heard earlier in Dr. Saeed's talk about some different characteristics of a medication that make it more prone to having difficulties with the generic switch. And I'm actually not sure it is does clobazam have some of those properties, Dr. Saeed?
Yeah, no, he didn't mention any sustained release properties for that. Okay. Here's another question from one of the audience. He said, if this is a question for the pharma companies, if they have a pre-knowledge about the drug and the effect and the exact concentrations and the component, why is the generic forms are variable? Is it just this is the way they make it, or is it just cheaper for them, or is just this is the nature of combining the medicine? I don't know if you, Dr. Yusuf bin Jardan or Dr. Lina.
Yes, I think the like manufacturing and then like manufacturing or the pharma industry, they have different parameters to keep in account. Usually the kind of the raw material they are using, it might change the pharmacokinetics profile and also the source of the material they have. So some like companies, they might looking for cheaper like raw material. So this kind of raw material, it might change the bioequivalence and bioavailability parameter. So having a good quality raw material will be advantage for the any generic company.
Okay, good. So this takes us to another question from the audience. What shall we do if we think the drug is not effective? Should we report this to the pharmacy? And I don't think this is an easy way. I mean, Dr. Johnson mentioned this in her talk as well, that number of seizures, breakthrough, and side effects. So just calling the drug is a failure or is not effective is a big thing. I think as a clinicians, we always have this conflict. Should we just go with the patient view and just believe everything he says, or especially the side effect, not the seizure breakthrough? So I don't know, Dr. Johnson, what do you think about this one?
Yeah, this is tough because as you pointed out, it's sometimes hard to know. And so far, I haven't noticed or I haven't observed that any particular generic manufacturers are worse in general, with the exception of the TIVA modified release ones where I pointed out those shells. But per patient, I just ask the pharmacy to please go back to their prior generic manufacturer if we thought that was better. Yeah, I don't think it's, unfortunately, I don't think it's something where we have a monitoring system to say, oh, this generic manufacturer has more problems than another. But that's a very interesting point.
Yeah. Okay. I personally had some bad experience with the sustained release form. I had one patient who switched it from the regular one to the sustained release form, and then he just had multiple seizures. So I had to switch him back to the regular one, and then it's kind of hard to gain their trust again. So any future drug modifications, just gonna take this with harder to convince them. Okay. Here's other questions from the audience. Seizures related to traumatic brain injury, which drug you prefer? Any special drug preference for pediatric versus adult? So maybe a clinical question. I don't know if you have experience with Dr. Johnson who can answer this question.
It's a good question. Just personally for brain injuries, I tend to like glucosamide and or Depakote, kind of based on what the patient can tolerate in terms of side effect profile. But that's just kind of a personal preference. Okay. Well, I received many comments thanking for the great speakers and educational material that all you provided. There is one general comment that I received many times, and I think this is just from a clinical perspective. They say we have some generic antiepileptic medicine that has more side effect or less clinical effect. However, they claim that the bioequivalence is the same. How could we explain this one? I think Dr. Johnson, you can answer this. I can try. I do think that it probably comes down to that broad range of acceptability that I talked about. So the confidence interval has to be between 80% to 125% of the brand. But you can imagine if a generic is down close to that 80%, it's probably giving you less active agent. So there's been some proposals, oh, should we tighten that window? Should it be 90 to 111% for neurotherapeutic index drugs? But I agree. I think there is possibility for problems, especially if they have that low, if they're towards the low end of that range. And if that's the case, I do like to check a level before I switch someone from a brand to generic. And then if they can't go back on the brand due to cost, then I'll just increase the generic. I'm not sure if other people have had that experience as well.
Yeah. So I was asked as well to be part of the committee to evaluate the different generic for levetiracetam in my hospitals. I looked into the evidence a few months ago, and there was one big studies in Italy. They compare the generic versus the brand name, and there were no clear differences between the switching. So I was wondering, is the newer antiepileptic medications, are the pharma companies are more smarter now in making the generic forms versus the older one, where we tend to have more problems? So Dr. Johnson or Dr. Yusuf? Yeah, Dr. Johnson, go ahead.
Sure. I know I've observed that too, that the newer ones seem to be easier. And I don't know if that's because they're the non-inducers or just other properties that make it easier to copy. But it seems like levetiracetam usually has a tighter confidence interval for the AUC and the Cmax. And also those modified release profiles that I showed you, they were much more close to the brand for levetiracetam. So I don't know if it's just easier in some ways, or if it's enzyme-inducing properties, or what. I'd love to hear Dr. Benjardin's opinion.
Yeah, Dr. Yusuf?
Like I totally agree with Dr. Johnson with what she said. So I believe it's like pharma industry is kind of complicated industry. So I think they are smarter, like in different way.
Okay. Here's the questions for Dr. Lina Saeed, if you can answer. Which pharmacological factors are more clinically important? The actual drug effect, the clinical effect, the Tmax, or the excretions? So which one of all these bioequivalence markers are more clinically in tuned, as we can say?
Speaking about the antiepileptic medications?
Yeah.
Actually, we cannot decide that this pharmacological parameter is more important than the other, because they are all in combination. They are making the final clinical picture for the patient. And what we are aiming for is the patient to be free from the seizure episodes. So once the patients reach this state, I don't think that it's not practical to measure the Cmax for a certain patient, and it's the area under the care for that patient. We're not doing this in real life, in practical life. We're just monitoring the patient for the number of seizures and reaching the free of seizure state. So I don't know. Maybe Dr. Yusuf can explain more on that.
Like I think the parameter is very important for various like regulatory affairs. So the numbers are the same, so they cannot judge. So I think it's up to the clinicians to see what's if he switch back and forth from the brand, he can decide which one is going to be a good choice for the patient themselves. So like I think what I heard about the like regulatory body, they are doing like sampling every time, and they are testing a lot of the generics. So if they see anything suspicious, they like contact the company to give any reasons if they change anything in formulations. And after that, they can just make a public announcement about any problem. But I believe, from my point of view, I think clinicians, they have the right to decide which one is going to be good for the patient.
Okay. Thank you. This is very good. So from clinical perspectives, I mean, as a general practitioner or someone who's dispensing medicine for a controlled patient, do you see the switch just because of the price as the driving factor, or because of the sustained release as a main driving force in your clinical practice, Dr. Johnson?
Yeah, so definitely cost is the main driving factor from switching someone to from a brand to generic. Even with insurance coverage, the brands can be thousands of dollars per year versus the generics, which will be much, much less. So cost definitely the main driver. When I tend to use the sustained release drugs, I tend to do it if I think a person is having side effects on the immediate release drug. I found that the modified release can often give a much better side effect profile. And I personally tend to still dose them twice a day because I'm not sure I trust the 24-hour coverage for all sustained release drugs. So I have a few patients where they can only manage to take drugs once a day, and then, okay, then we use sustained release. But I've had a lot more success probably in terms of side effect control rather than definitely seizure control, switching from immediate to modified release.
Okay, and typically in your first follow-up after switching patient from brand to generic, what are the first things that you ask your patient about?
Well, of course, first, any seizures to report, of course, but then I tend to get into the side effects too and see if they've had any, noticed any other changes.
Okay, and how do you deal with patients if they're just, okay, I'm not happy, I want to go back to this medicine, and really there are no objective concern from the patient? This is a common scenario for all of us as a practitioner.
Not happy on the generic and they want to go back to brand?
Yeah.
Got it. I say, well, I can prescribe it, but the insurance is probably not going to cover it. I don't think if I fill out a paperwork that says patient not happy, that the insurance would cover it. This might be a difference in our healthcare systems because I actually haven't encountered that very much at all, just because the patients know how expensive it will be. You said that's a somewhat common problem.
Okay, yeah. So thank you very much, Dr. Johnson. I have no further questions. I don't know if Dr. Lena has any comments or Dr. Yusuf has any comment.
Thank you very much.
Thank you very much for having me. It's my pleasure to be one of your presenters. Thank you so much, Dr. Majid and Dr. Emily and Lena as well, and for the team for this great webinar.
And thank you all for this invitation and this great discussion. This is really fun. Thank you.
Okay, thank you all for the great educational materials and discussions and for your time and commitment. Thank you very much. Okay, bye-bye.
Bye-bye.